Today we're going to talk about sequencing DNA and the Human Genome Project. So first, I want to talk about the Human Genome Project. It's like 1984-ish to like 2003. All right. So, have you ever heard of that before? Human Genome Project? What was the point of the Human Genome Project? Anybody know? Yeah, map out the DNA. Map out all the A's, C's, G's, and T's of the human DNA. So, the purpose was to map all the A's, C's, G's, and T's of the human DNA sequence. Okay. Now, this process took a really long time. It started in 1984, and their original plan was, okay, if we can figure out the human genome, we can, look at all the proteins that we can make, and we can figure out exactly where in our genome those proteins are made. And so if somebody has a genetic disorder that stops them from being able to make a protein, we could fix it by knowing where in the genome that problem occurred. Okay. So in 1984, they came up with this idea to be able to sequence the human genome, and we had sequenced genes before, and we'll talk about how we actually went about doing that. But we started planning this project in 1984. Then in 1994, 10 years later, they had sequenced something like 5% of it in 10 years, so that wasn't very good. Right, and then over the next couple of years, they did a little bit better. They sequenced like another 20% in the next 4 years, so picking things up. And then in 1998, there was this other company, and so this is all, it was like international governments and stuff like that, and a lot of money was put into this. I think it was like $2 billion total, and that's actually not that much money when you really think about it. But $2 billion was put into this project. And then there was this other company called Celera Technologies. And Celera Technologies, I don't know if you've ever had blood work or anything done, but there's a company called Quest Diagnostics where you'd go to get your blood work done. Quest Diagnostics is owned by Celera Technologies. And they were like, hey, I bet this gene sequencing thing is going to be the new hot thing to do in medical technology. So let's just figure out our own way to do it. And in 1998, they started their own Human Genome Project, and they finished theirs in 2003 when the Human Genome Project, this one, wasn't even anywhere close to finishing. And they were like, hey, we're done. What about you guys? And so they stopped what they were doing. They were like, okay, we won't work on that anymore. Because Celera, being a large, corporate company that had all kinds of technology at their fingertips, they didn't go through it in the normal sort of manpower way. They went through it in the, we have a lot of technology and we're going to use computers and lasers and things like that to find a better way to do it, a lot faster way to do it. So they were able to complete theirs in 5 years, which is pretty impressive. In 2003, they were like, haha, we're done, and we're not going to tell you guys, neener. And there was like this huge international backlash against them, and so finally they did agree to release what they'd figured out and how they did it and stuff like that, but not without a little bit of angst and begrudgingness. So let's talk about what they figured out in the Human Genome Project here. First of all, number one, the original intent was to determine which genes made which proteins, to pair genes with proteins. The proteins they produced. Okay? What they found was a little different than that. It was a lot more confusing. What they found was that the human genome is about 3 billion nucleotides long. And of those 3 billion nucleotides, they found that about 1.5% of them code for proteins. So I want you to take a step back for a second and think about this. So for the last 3 weeks, we've been learning all about the very important process of protein synthesis, whereby DNA is transcribed into RNA, and the RNA is sent out into the cytoplasm. That RNA is then translated into proteins, and we said that that's how genetic information is stored and passed along, and that's what makes you you and all kinds of stuff like that, right? Seems important, right? 1.5% of your genome does that. The other 98.5% does something else. And the real messed up part is we're not real sure what it does. For instance, one of the very strange things is if 1.5% are coding for proteins, 75% of the genome gets transcribed. Okay, so that means it's turning into some kind of RNA. Makes some form of RNA. So we know, for instance, there's like rRNA, that's the stuff that ribosomes are made out of. There's tRNA, right? That's the stuff that's actually doing the translating. So some of that has to be in the DNA, but a lot of it is not. A lot of the part that is the 75% that's being transcribed is not rRNA or tRNA. A lot of it is this kind of RNA that's called microRNA. And microRNA, we know, has a very large part in which genes gets transcribed and how often they get transcribed and things like that. But we don't really understand the mechanism behind it, right? In fact, there are hundreds, perhaps thousands of other types of RNA that are produced that we just have no idea what they do. That they're present in living things and all living things, and we cannot figure out their function. This is what you should be excited about. I won't tell you to get excited too many times this year, but these kinds of things, whenever I tell you that we just plain don't know something, especially something as important as like the stuff that's in every single one of our cells that carries our genetic information and we don't know what it does, that is a big deal. So a lot of people I'll talk to and say like, oh, what do you want to do when you grow up? I want to like find a cure for cancer or something like that. Okay, cool. But that's not just going to happen. Like you're not just going to be messing around in a lab and be like, Eureka, I've cured cancer. It's all going to come from understanding, right? It's going to come from understanding as to like why these cells become cancerous in the first place and what natural resistances we have to cancer and how we can implement those natural resistances in people who have lost that ability, right? And the key to all that is going to come from this, right? It's going to come from figuring out how the rest of our genome is actually interacting and figuring out what this remaining 73.5% is doing that's getting transcribed. We know a lot of our genome does kind of weird stuff, like non-genetic stuff. Specifically, we know that around 44% of it is repeating segments. 44%, or rather, are repeating segments. And we know a couple of those repeating segments. We talked about them already. There's a kind that's called a telomere. Okay, those are the ends of the chromosome. We know that telomeres are necessary because every time DNA replication happens in a linear chromosome, like eukaryotes have, that it degrades the end a little bit. You lose a little bit of the chromosome, you lose a little bit of the DNA off the end just because of the process of DNA replication and how you have to have a primer and everything like that. You lose a couple nucleotides every time it needs to be copied. And so every time the cell then divides, the chromosome gets shorter and shorter and shorter. And for a long time, and kind of still, we think that that has something to do with this process that's called senescence. Senescence means that cells will die of old age, right? So they can't divide indefinitely. We talked about in here the line of cells that we do almost all experimentation on in labs, the line of human cells called HeLa cells. They came from that lady named Henrietta Lacks. Henrietta Lacks had cervical cancer. They took some of her cervical cancer cells. Her cervical cancer cells happened to have a mutation in a gene for telomerase, right, which is the enzyme that builds more telomeres, and it's generally only active in embryonic cells, but hers became active in these cancer cells, and so they just keep growing. They can divide indefinitely because they repair their own telomeres as they divide, which is crazy, right? We know that there are cells that don't divide very often that still die of senescence, so we know it's not the only thing that controls the number of cell divisions that a cell can go through, but it's one of the things, okay? We know that there are these things called centromeres. Centromeres are where the sister chromatids attach, right? And then we know that there are these just long interspersed repeats and tandem repeats. And these are largely misunderstood or not understood. It's not that we say that we know what they do, we just don't know. We don't know their function. So, tandem repeats, interspersed repeats, these are just sequences of DNA that tend to, be maybe 1, 2, 3 nucleotides being repeated over and over and over and over and over and over again in the middles of genes sometimes between genes, in various spots for whatever reason. Okay? So, lots of stuff that we don't understand. Next, I want to talk about this original process of sequencing the genome, and we're going to go through and sequence a DNA sequence. We're going to try to figure out the order of a DNA sequence using the same method that was originally used. Okay, so let's do DNA sequencing. It's discovering order of A, C, G, and T in a sample of DNA. Okay, so normally in the lab they would do this process with a DNA sample that was, let's say, 200 to 300 nucleotides long. That would take us a really long time to do that, so we're not going to do that. We're going to do 15 nucleotides. So I'm just going to throw 15 nucleotides out here pretty randomly. Let's say G, C, G, A, T, C, G, A, C, T, A, C, T. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, C, A. Okay, so 15 nucleotides here. This is what we're going to figure out the sequence of. And the way that we're going to do it is we are going to use the polymerase chain reaction, right? PCR. And so in this PCR, we are going to do something slightly different though. Instead of putting in a bunch of free nucleotides, we are going to put in a bunch of free nucleotides, but we're also going to take one of the nucleotides, let's say thymine, and we are going to put in a defective form of thymine. So let's say that 5% of the nucleotides that we put in are defective thymine. So I'll do T star to represent the defective thymine. Now, the defective thymine is a thymine molecule that's called 2', 3' dideoxynucleic acid, or deoxyribonucleic acid. Now, that sort of matters, and you sort of understand what that means already. It is in contrast to deoxyribonucleic acid, which is the normal kind, which its actual name is 2' deoxyribonucleic acid. And the reason why it's 2' is because it says that on that second carbon, there's no oxygen. It's deoxy, right? So that's where it's losing the oxygen. And so you guys know, hopefully, you guys know that a dehydration reaction, which is the reaction that joins two things together, is going to require an H group and an OH group, right? The H group is on the phosphate side, and the OH group is bonded to the 3' end, and that's why when we talk about the 3' end of DNA, we always say that's where the bond occurs, because that's where the hydroxyl group is present in that molecule. And the 2' doesn't have that. Well, in this funky thymine here, you've got no hydroxyl group on 3 and no hydroxyl group on 2, and that means it can't bond to anything else after that. Okay? So let's say this sequence goes along, and the first one that it creates is this sequence. It goes through and it says C, G, C, T, and let's say this is an OK thymine. That's a normal thymine, one of the 95% that we put in. A, G, C, T, there's another normal thymine. G, and then let's say this, oopsies, A, and then this thymine is the one that's messed up. Okay, so a messed up thymine, a defective thymine bonds in that spot right there. That means that the rest of the sequence can no longer continue because we can't bond anything to this di-deoxyribonucleic acid. Okay? PCR goes on again. Do it again. C, G, C, T. Let's say this one is the defective one now. So we can't make anything longer than this. Okay? Next one, C, G, C, T, A, G, C. Here's the defective one. And the next one, C, G, C, T, A, G, C, T, G, A, T, G, A, G, T. Here's the defective one. Okay, so these are the only 4 molecules that can be made in this PCR, right? Because we have 1, 2, 3, 4 adenines, and these are the 4 different spots where the defective thymines could go to, right? Yes? Everybody following so far? Have I lost anybody? Does anybody have questions? All right, we're going to take this, we're going to take these 3 segments here, and we are going to add them to a gel electrophoresis plate. Okay? This gel electrophoresis plate is going to have 5 chambers in it. Okay? The first chamber is a chamber that is used as a reference point. Okay, so let's say in the first chamber they put in a nucleotide sequence that is 5 nucleotides long, one that's 10 nucleotides long, one that's 15, and one that's 20 or something like that. Okay, and so then that will run through and it'll say, okay, the one that is 5 nucleotides long will end up all the way down here. The one that is 10 nucleotides long, let's say, ends up here. The one that's 15 nucleotides long ends up here. Okay, so they know that this is 5, this is 10, this is 15. Okay? So then in between there, they can put little markers and say this is 14, 13, 12, 11. This is 9, 8, 7, 6. And this is 4, 3, 2, something like that. Okay? Everybody get what's happening so far? All right. They take this stuff from the PCR, and they put that into that well right there. Okay, and we made a couple of different segments. This first segment was 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 nucleotides long. This one was 4 nucleotides long. This one was 1, 2, 3, 4, 5, 6, 7, 8 nucleotides long. And this one was 15 nucleotides long. Okay, so one of the bands is going to be right here. One of the bands is going to be right here. One of the bands will be right here. And one of the bands will be right here. Okay, so what does this tell them then? This tells them, based on using their reference point here, that one of the nucleotide sequences that they created was 15 nucleotides long. And that tells them that there is a nucleotide that bonds to thymine at the 15th nucleotide. So the nucleotide that bonds to thymine is adenine. So they know the 15th nucleotide in the sequence is adenine, right? They know that there's a strand that's 11 nucleotides long, which tells them that the 11th nucleotide will be adenine, right? There's one at the 8th that tells them that there is an 8 nucleotide adenine, and then one at 4. So this is 4, 8, 11, and 15, right? They take the same process, and instead of putting in defective thymine, they put in defective adenine, and that tells them the locations of the thymines, right? Then they use defective guanine, and that tells them the locations of the cytosines. And then they use defective cytosine, and that tells them the locations of the guanines. So they'll end up with a gel electrophoresis plate where they put in like the defective thymine is there, the defective adenine is here, the defective guanine would be here, and the defective cytosine would be here. And there will be a line at every single mark here. And that's how they know, okay, this mark represents this nucleotide, and this nucleotide bonded to this defective nucleotide. Right? This doesn't seem overly complicated, right? This seems like if you've given enough time, you might have been able to come up with something like this to figure it out on your own, right? It's not like ridiculous. But it takes a really long time. We did 15 nucleotides here. They do sequences that were like 200 to 300, and the nucleotide sequence is 3 billion nucleotides long. So that takes a long time. And then also, you have to consider the fact that you don't know which segment you're looking at, right? So sometimes you are doing a segment of DNA here that's 300 nucleotides long, and you don't know where that came from in the nucleotide sequence. And so you have to go through and put it into a computer and have the computer say, okay, well, the last 80 nucleotides of this segment overlaps with the first 80 nucleotides of this segment, so they piece together right here. Right? And that's how we figured out the sequence of nucleotides. Took a long time. They won a Nobel Prize for it, though, for this. Yeah?
Wait, so that was like the faster way?
No, this was the slower way. The faster way, this was the original Human Genome Project. The faster way was that Craig Venter, when his company Celera Technologies went through, and instead of just putting in defective thymines, defective adenines, and guanines, and cytosines, he put in defective ones that were made with a phosphorescent marker that made them essentially respond to a certain wavelength of laser light. And then instead of doing gel electrophoresis like this, he suspended them in this tube, this glass tube called a capillary, where they would suspend based on how large the length or how large of the molecule was. And then there was a little laser, 4 little lasers that would shine, and it would take a picture, and it would move up like a micrometer and shine and take a picture and move up and move up and move up. And it would then be able to see based on which lasers made the nucleotide glow, which nucleotide was at that spot. And so it did the same process, but allowed a computer to do it rather than humans. And it went wildly faster. Now, I always have to go through when I do this unit, and I have to kind of touch up on what the current technology is, because it'll blow your mind. So first of all, we know that the original project took a really long time. 10 years didn't really get them very far. They had like 5% done in 10 years. Then Celera Technologies came along, did one nucleotide sequence in 5 years. Okay. Did I already talk about the okay, I don't think I have in this class. The Human Genome Project is 3 people. Like, not 3 people doing it, it's the genomes of 3 people. So, like, it's not really the Human Genome Project, it's like the specific humans genome project, right? Because everybody's got different DNA. There's another project now that's called the 1000 Genome Project. And the 1000 Genome Project is exactly what it sounds like. It's get 1,000 genomes and get it sequenced. And that was in 2008. I think they finished that in 2012. And now that same project is going on, but it's kind of like the as many genomes as we can get our hands on project. It's got like 2,500 genomes in it right now. And they're doing a really good job of getting people from different descents. So they have like people of Asian descent and of European descent and of African descent and South American descent and stuff like that. So they're able to determine a lot of cool things about like ancestry and historical events get tied in to say like, hey, look, there's a lot of Spanish DNA that shows up in Central America. Wonder why that is. And they're like, oh, wait, now we know that is. So a lot of cool things are happening like that. But the most mind-blowing thing, and the reason why that project can exist, is that the current technology sequences the entire human genome in 3 hours. And it doesn't take a lab to do it. It is a little machine that's about the size of a microwave and could sit on my desk, and you just like pop the DNA into it, and it like does its thing and spits out the nucleotide sequence for you. Yeah.
I was gonna ask like how they chose the 3 people. Did they like disclose?
I believe 2 of them were working on the project, and one of them was James Watson, of Watson and Crick, the guy who discovered the DNA helix and the structure. What's that?
No, not me. I was thinking there's a lot of James Watson. There's like James B. Watson.
Maybe. I'm guessing there are. It's a pretty common last name. All right. So the last thing that I'm going to do, I'm going to show you an advertisement.